Hi, my name is Andre, and my metabolic disruptor is glucose-6-phosphate dehydrogenase deficiency. To begin, I'll explain the metabolic pathway that's involved. Pentose phosphate pathway is an anabolic pathway with two phases, an oxidative phase that creates NADPH and a non-oxidative phase that generates pentose sugars. This process occurs in the cytoplasm of human cells. This pathway is also called the pentose pathway shunt because it shunts carbon back into the glycolysis pathway or the gluconeogenesis pathway, depending on the organism. Now, additionally, it has ties to the glycolysis pathway because it occurs parallel to glycolysis. Now, as opposed to glycolysis that generates ATP, the end products are a multitude of intermediate precursors for future synthesis. It does not involve ATP, however, it can be used as a detour inside glycolysis. As we look to our figure on the right, glucose-6-phosphate has the option of whether it can enter PPP or the citric acid cycle depending on the need of the cell. If it needs energy from ATP or electrons from NADPH. Now, depending on the organism, the metabolite can be fed into many pathways, and the main enzyme that catalyzes PPP are primarily located in the liver. So why is it important? Well, it's important for a number of reasons. It generates NADPH, which is the most important molecule for anabolic processes. It provides various precursors for many nucleotide amino acid syntheses, such as ribose 5-phosphate for nucleic acid synthesis and erythrose 4-phosphate for aromatic amino acid synthesis, to name a few. It also provides reducing elements to other pathways that might need it, and it keeps cells loaded with antioxidants to combat oxidative stress. As to why it's relevant and interesting, the pentose phosphate pathway has a huge role in cancer cells. PPP is a huge contributor to cancer cell survival and growth, and it provides NADPH to the cancer cells while they're under oxidative stress. The cancer cells also use NADPH for fatty acid synthesis, DNA damage response, and other purposes. So it seems like PPP is a bad guy, but it can be used in our advantage. A way of combating these cancer cells is to target certain enzymes with PPP to be used as cancer therapy. Now, as for the biochemistry of PPP, the first phase is the oxidative phase, which is an irreversible phase that oxidizes a glucose-6-phosphate into a ribose 6-phosphate, while in the process generates two NADPH molecules, carbon dioxide, and two H+ ions. With the help of a few intermediates and enzymes, the enzymes help catalyze the respective intermediates to go through processes. On the other hand, the non-oxidative phase is a reversible phase, and it can be used to convert 3 molecules of ribose 5-phosphate into 2 molecules of fructose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. With the help of enzymes such as transketolose and transaldolose to convert intermediates such as xylulose 5-phosphate and ribose 5-phosphate into the molecules just mentioned. These components are then sent on to glycolysis. Now, as for the regulation of PPP, there's a few ways it can be regulated and many more. The first one is a flux of PPP in the production of NADPH can be increased through transcriptional activation of glucose-6-phosphate dehydrogenase from P73, which is a protein that has tumor suppressive functions. Also, cyclin D3-CDK6 is a complex that inhibits PFKP, which is phosphofructokinase platelet, in cancer. When this occurs, the flux of glucose is altered in PPP. This leads to more survival mechanisms as opposed to storage mechanisms when under stress. Now, the deficiency of glucose-6-phosphate dehydrogenase was first discovered in the 1800s in Greece, Italy, and Portugal, where doctors noticed severe anemia and hemoglobinuria in children. It was not given a name until decades later, but it was first deemed as favism, which were severe reactions from consuming fava beans. It is the first enzyme of the PPP and is also the rate-limiting step. So inhibiting this enzyme will have a domino effect on the rest of the pathway. G6PD provides reducing power to many cells in the form of NADPH. Without this enzyme, the production of NADPH increases and can eventually lead to the increase of reactive oxygen species that can lead to red blood cells being damaged. Now, in humans, G6PD deficiency can cause hemolactic anemia. Hemolactic anemia is a blood disorder where red blood cells are destroyed faster than they are created. Symptoms of hemolactic anemia include enlarged liver due to the excess of damaged red blood cells, jaundice, a condition that causes whites of eyes to turn yellow, and a faster heartbeat and shortness of breath due to lack of oxygen circulating from red blood cells. Now, treatment of G6PD deficiency, sadly, it is an X-linked recessive disorder, so there is no cure to this deficiency. However, the best way to cure G6PD deficiency is by avoiding any sort of oxidative stressors. These can include excess alcohol, high levels of stress, and lack of sleep. All of these can trigger the deficiency and can lead to oxidative stress. There's also a lack of information to prove if antioxidants such as vitamin E and selenium are beneficial for treatment. However, research is being done on finding medication to hopefully combat this deficiency. A dry extract from Pinus pinaster called pycnogenol is currently being tested for a possible option of treatment. It has been said to have good ability to scavenge oxidative agents. And that concludes my presentation.